plant genetic resources. In this session, we will be having 3 presentations from the division of biochemistry, molecular biology and biotechnology, and plant physiology. I request Dean and Joint Director Education IARI, Dr. Rashmi Agarwal, to formally welcome the chairman and the audience.
Good evening, all of you, and once again, welcome all of you in this auditorium on the fourth session of the presentation of significant educational achievements in the School of Basic Sciences. So we are honored that with us we have today the chairperson of this session, Dr. K.C. Bansal, former director, NBPGR, and a very renowned biotechnologist. First of all, I would like to request our director, Dr. A.K. Singh, to welcome the chairperson with a bouquet of flowers. Friends, we had seen 3 sessions before this session. Morning, before lunch, we had 2 sessions. One was crop improvement, and then followed by crop protection. Post lunch, we just finished School of Natural Resource Management, and now we have School of Basic Sciences. I welcome on behalf of PG School and on my own behalf our director, Dr. A.K. Singh, who will be there guiding us throughout the session. In this session, ladies and gentlemen, we have 25 thesis presentations, out of which 15 are MSc students theses and 10 are PhD students, which covers biochemistry division, molecular biology and biotechnology, and plant physiology. So the professors of the respective disciplines will be giving their presentations. So I once again with these words welcome all the off-dais dignitaries. PD Dr. Man Singh, if he is here, he was here beforehand. I would like to also welcome Dr. D.P. Ray, who is the chairperson in the next following session of School of Horticultural Sciences. So we welcome you to this presentation of the School of Basic Sciences. I would like to welcome all the heads of the division, professors, faculty, students, ladies and gentlemen. So we are going to listen to the performance of the students under School of Basic Sciences. Thank you very much.
I request Director IRI, Dr. A.K. Singh, to briefly introduce the chairman of this session.
Very good evening to all of you. It's indeed a great pleasure for me to welcome one of our great colleague, Dr. K.C. Bansal. I had an opportunity to be, as a student in Sisir Hostel when he was about to finish his PhD program when we had joined in 1986. He was working with Professor Sinha, and he had a lot of interest in genetics, understanding the physiological basis of many of the wild relatives of wheat, and particularly for biotic, abiotic stress, abiotic stress tolerance. He will come and discuss a lot on the stability analysis, more of genetics than physiology. Always he has been one person who is always full of energy, enthusiasm, and he infects the energy and enthusiasm in the person who works with him. For last say about 1 week we have been working together on the genome edited crops guidelines for the deregulation. There is a draft guideline which has been proposed by DBT, which is open for comment. Now it is extended to 23rd of February. Earlier it was 8th of February. But the Academy conducted a brainstorming session which Dr. Bansal is leading, and many very good ideas have come. He is working day in and out, and anytime I open WhatsApp, there is something to suggest, to correct, to incorporate. So that's the kind of energy with which he works. He is as Director of National Bureau of Plant Genetic Resources. He did a commendable job of taking out almost 25,000 wheat accessions, getting them evaluated for biotic stresses, in particular at Wellington Station and also at Haryana Agriculture University. So this was ever for the first time such a large number of plant genetic resources were taken out from the gene bank, evaluated, and many potential germplasm lines were identified. Good publication came out of it, and now the program is being followed in other crops also. And now again he is involved with the Department of Biotechnology, leading a major project in wheat, particularly characterizing the plant genetic resources in wheat for their potential utility in the breeding program. I have been fortunate in particular to work with him for very long on several forums, in several committees, and as a good friend. As a human being, he is a great person. Whenever you are in difficulty of any kind, personal difficulty, he will take care as his own family member, brother, whatever you can say. I had at least 2-3 such experiences. Once I suffered from dengue, and there was a lot of difficulty in getting admission in Gangaram Hospital. We were left out 8 hours, thrown out of emergency, and it was Dr. Bansal who came for rescue with his some acquaintance. He found a place in some hospital, Agrasen hospital, and then I could be saved. So whenever it comes, at personal level also, he is a great friend. It's my pleasure to welcome him for this presentation of basic sciences, and we look forward for very constructive suggestions from him. He is academically very well achieved person. He has been postdoctoral fellow at Harvard and made excellent publications in high rated journals, including PNAS. And as a teacher, and then pioneering work that he has done in the transgenic research, identified for the first time genes for abiotic stress tolerance, which have been deployed in several breeding programs. It's my pleasure to welcome you, sir, and thank you for accepting our invitation.
Thank you, sir. I now request the convener of the session for further proceedings.
Very good afternoon, everyone. Respected Chairman Dr. K.C. Bansal, Director IARI Dr. A.K. Singh, Dean Ma'am, dignitaries of the dais, dear faculty and dear students. Ma'am has already discussed, so I will not waste time in mentioning which schools are presenting here. Just a simple request to all the 3 speakers, just please maintain time so that we don't get much more delayed for the next session. And now request Dr. K.C. Bansal to start the proceedings.
Thank you, Neera. Thank you, PG School. And before I go into the proceedings of this particular session on basic sciences, I would like to convey my gratitude and thanks to the previous Director IRI, Deputy Director General Extension, Dr. A.K. Singh, and Dr. Rashmi Agarwal for inviting me to this particular session to interact with you, particularly in the area of basic sciences. I'm extremely delighted to come to this Convocation Week celebrations of the great institution we have in our country, the greatest in Asia, or in fact, if you think many parts of the world, Indian Agricultural Research Institute. And when it happens today on 11th February of 2020, and particularly I'm so delighted that we have Dr. Ashok Singh as our new current director of this great institution. And thanks to him that he introduced me so nicely that I enjoyed listening to you myself, I must admit it. But at the same time, when we were talking professionally, when I was here all through my life almost, right from 1983 onwards, and he mentioned about my PhD work, which I was doing with Professor Sinha, I must tell you, I have always gained and continue gaining from the valued experience which Professor Ashok Singh brings with him. And therefore, I'm sure all of us are very pleased. In fact, I'm still not fully recovered from the happiness which I have, really speaking, witnessed myself within me. I continue still being more and more happy day by day that yes, a person like of his stature, Dr. A.K. Singh, is the director today of this institution. I want to express my gratitude here before you all, particularly to the students. And Dr. A.K. Singh has been, as you all know, I know personally that whatever I know little today about genetics or plant breeding, it's all because of his, teachings to me. I will walk to his room, he will give you a nice cup of tea, and more than that, he will take your class. He will teach you very nicely the basics. So he's been a wonderful researcher by himself, a great scientist, an excellent teacher, and above all, a very nice human being. And more importantly, for an institution like this, if you're heading, you must be interacting with the students directly. And he's very popular among students, I know this. And more importantly, again, when you talk of product dissemination or any knowledge which we produce here in this institution has to be finally for the use of the welfare of farmers. He is one person whom I always talk about among my friends. He is one who is very well known among farmers as well. You would have seen it, you will see it more. And what he means only the business, he means only what we can do for this country, including, whatever through agricultural sciences. And I'm very happy. This was the first opportunity I got, actually speaking, that I'm visiting IARI in a formal capacity, and I'm able to talk to the audience like this, who has been wonderful students of IARI, the faculty members and others, and that I thought I should express myself, though I've taken a few minutes extra out of the schedule, and I'm sure you will kindly excuse me for this and adjust as we proceed forward. And I'm very happy that we have in the audience Professor D.P. Ray, the former Vice Chancellor of OUAT Orissa, Professor N.K. Singh, most learned, biotechnologist and genomic scientist and expert in this country, internationally renowned. And many other of you are actually speaking. Those professors going to make presentations are all my very good friends, head of the divisions and the professors. And I, of course, always happy, to interact with the students more and more. So with this, once again, I thank you all, friends, for giving me this opportunity. And without taking further, time on these things, I would now proceed further with the presentations of the professors. And the time has been allotted to you each. And I'm very happy the time has been allotted as per the number of students they're going to talk about the research. We have, in case of biochemistry, 10 minutes, molecular biology and biotechnology 18 minutes, plant physiology 13 minutes. And I'm sure I would request each professor to kindly stick to the time, because that's very important. And that's one of the lessons we may like to teach the students as well through the time allotted to us, what presentation and what good presentation we may try to make. That's also one of the part for our students to learn through our professors. So therefore, to begin with, I have a pleasure inviting Dr. Professor Anil Dahuja, the professor of biochemistry, to make a presentation for 10 minutes on the achievement of the students' work which must have been done, in the previous year in the division of biochemistry. Thank you, friends. Yeah, Dr. Dahuja, please.
So, Honorable Chairperson of the session, Dr. K.C. Bansal, respected Director of the Institute, Dr. A.K. Singh, Dean Ma'am, and dignitaries of the dais. So, it is my proud privilege to be presenting the significant educational achievements of the students of the discipline of biochemistry in front of you. So, we have a very small faculty. Total, we are 11 faculty members. Out of that, 9 are eligible for guiding the PhD students. And student strength at present, it is MSc 9 students are there and PhD 15 students. And trimester wise, we have breakup of various courses like this. So then the major thrust areas in which the division of biochemistry is working is the nutritional and keeping quality enhancement of the food crops, and then abiotic stress tolerance. So this year we have 3 MSc students who are working on the first area, that is the nutritional quality enhancement. 1 student worked on the soybean, and the 2 students they work on the pearl millet crop. While in abiotic stress tolerance, we are working on the wheat and rice both, but this year one MSc student worked on the wheat. So total 4 MSc students, they will be getting the degree in the forthcoming convocation. So in the nutritional quality enhancement of the soybean, the problem which has been addressed is the low micronutrient bioavailability due to the presence of phytic acid in soybean. So you see, this is the structure of the phytic acid, which is containing the 6 phosphate group, and because of the presence of this negatively charged phosphate group, it is able to chelate the metal ions and thereby reducing their bioavailability. So this is how the phytic acid is actually leading to the deficiency of the minerals due to its presence, and this is how the phytic acid is getting synthesized. So there are 2 pathways by which it can be synthesized: lipid dependent pathway and the lipid independent pathway. And here you can see that once it is synthesized, it is getting stored in the vacuoles due to the presence of the ABC transporters. So if we can somehow stop the transport of the phytic acid into the vacuole, so its accumulation, it can be prevented. So this is what the student worked on. So he targeted this ABC transporters, which is multi-drug resistant transporters. And the Smruti Rekha Sahu, she is the student who worked under the guidance of Dr. Archana Sajdev on the topic, the CRISPR-Cas9 mediated targeted genome modification for inactivating phytic acid transporter gene. And these were the 2 objectives: the identification of the gRNA and then development of the constructs and in vivo validation of these constructs in the aggregate method. And with regard to the objective 1, so they first selected the targets using various in silico tools, and then screened these selected targets, whether they were having the off targets or not, and then they studied the stability of these gRNAs. And this is how it is actually done. While the selection of the topics of this gRNA sequences, target sequences, they took care of these parameters, whether they were having the PAM sequence or the G was present at the 5' end or not, and all these parameters, they was taken into the consideration while selecting the target. So 3 targets they could identify, and then they looked into the structure of these 3 target sequences, and what they could find was that target 3, it was having the minimum free energy. That means it is having the more stability. So they worked on that. So this slide, it shows how this software, it showed that it is having the minimum free energy. And then with regard to the objective 2, they worked on the cloning of this gRNA sequence into the shuttle vector, and then cloning into the destination vector, and finally in vivo validation using the aggregate method, and finally the sequencing of the transformants. So this is how the blue vector, it was used as the shuttle vector for the cloning of the gRNA sequence, and then the cloning, it was confirmed by restriction analysis and also by PCR cloning and the sequencing. And then this was cloned into the destination vector, that is the MDC123 vector. Again, the cloning, it was confirmed using restriction analysis and the PCR amplification as well as the sequencing. So then they did the in vivo validation using the aggregate method, which has been standardized in our lab only. And then the sequence confirmation of this clone, it showed that there was a 1 insertion, and because of this insertion, you can see here this insertion, it has been identified. So there is change in the amino acid sequence, and that led to the confirmation changes in the structure of the protein, and that in turn led to the inactivation of the transporter. So in conclusion, they successfully designed and developed the Cas9 plasmid, and then they validated the integration. And the future, they are planning to use these constructs for the development of the low phytate soybean crops. Of course, you will be requiring more constructs targeting different regions of the same gene. So that will provide the extra window for the gene editing. So this was with respect to the first student. And then, as I mentioned, the other parameter which we were addressing was the keeping quality enhancement of the pearl millet. So in this, the problem addressed was the poor storage life of the pearl millet 4. So as you all know, the millet, it is a rich source of the nutrients, but because of its poor quality of the flour, it is not being used in the multigrain atta. So this is the problem which has been addressed, and they, after searching the literature, the students, they found that the major culprit for generation of, for reducing the quality of the flour, it is the lipid present in the seeds, and they found that this 5 to 7% lipids, they are present, and they are acted upon by the enzymes like lipase, lipoxygenase, and the peroxidase, that in turn is leading to the formation of the aldehydes and the ketones, which are actually causing the rancidity. So in lab they have standardized the methods, the comprehensive acid value and the comprehensive peroxide value, which is being used as the indicator for the quality of the pearl millet flow. So the student Durgasri Venkata Bhargav, he worked under the guidance of Dr. Shelly Parveen, and they targeted the first enzyme which I showed at the last slide, the lipase enzyme. So they did the in silico study, and then they used different methods for inactivation of this lipase enzyme. So when they did the in silico analysis, the one observation which was very enlightening was that this is having the very high aliphatic amino acid index, and the GRAVI, that is the grand average of the hydropathy, it is very high. So that means it is highly thermotolerant. So you require harsh condition to inactivate this enzyme. So then they tried different treatments on different varieties of the pearl millet. So these three varieties they were used. The WG100, which is the white variety, then Pusa purple one, which is purple one, and Dhan Shakti, which is a great variety. So these three varieties they were used in the study, and they did the biochemical analysis of these three varieties, and then they tried these three different treatments: the vacuum treatment, the decortication, and the microwave treatment, and then they studied the effect of these treatments on the lipase activity and the quality of the flow, which is indicated by the comprehensive acid value. So after removing the pericarp, that is a decortication, so they found that the lipase activity, it was not affected much. So only 6% change was there, and also because of the decortication, the loss of nutrient, it was there, because most of the nutrients, they are present in the pericarp region. So the reasons that could be possible for the less inactivation of the lipase could be that, so when tissue is disrupted, so it may lead to the enhancement in the lipase activity, as happens in most of the hydrolytic enzymes. So similarly, the vacuum packaging treatment they have used, and in this they couldn't find much of the reduction in the activity of the lipase because under the vacuum condition oxygen availability is less, so the anaerobic bacteria containing the lipases, they get activated. And similarly for microwave treatment, it was found to be giving the good effects because they could found the decrease of 26% in the lipase activity after the microwave treatment, and these were the conclusions which have been listed. So purple variety, it is the best variety in terms of the lipase and the comprehensive acid value, because this purple variety, it is rich in the flavonoids, so which is acting as the inhibitor of the lipase activity. And vacuum packaging, although it is not effective for the lipolytic activity, but it is very effective as far as the oxidative damage of the pearl millet is concerned. And then other student worked on the second enzyme, that is the lipoxygenase, and in this this enzyme, which is actually responsible for the peroxidation of the fatty acids. So same kind of varieties and same kinds of treatments they were used, and they studied the effect on the LOX activity. And here again they did the in silico analysis of the lipoxygenase enzyme, and then did the biochemical characterization, and then different treatments they were used, and they could find that in this case the vacuum treatment, it was found to be more effective as compared to the decortication and the decortication and the microwave treatment, because in this case the lipoxygenase uses the oxygen as the substrate. So when we are doing the vacuum treatment, that oxygen, it is not available, and that is causing the inactivation of the lipoxygenase enzyme. And then they also studied the effect of the combined treatment of the microwave and the vacuum, and this was found to be better than the individual vacuum treatment. So these are the salient findings of that study. And based on these studies, they could prepare a rancidity matrix, and that is now being used for classification of the various pearl millet genotypes into the different categories. So last student worked on in the abiotic stress tolerance area, and they were working on the Mitogen-Activated protein kinase enzyme. So as you all know that the MAP kinases, they are regulate many kinds of development and physiological responses by acting as a signaling molecule. So the student, and under the guidance of the Dr. Ranjit Ranjan Kumar, he did the he starts treatment at 30 degrees centigrade for 2 hours at the pollination stage as well as the grain filling stage, and then they did the analysis, expression analysis was done. and this is what they could find, that expression and activity assay, it was more the activity it was more during the grain filling stage in case of
You can just
thermotolerant.
you can conclude this particular thesis presentation with final conclusions, please.
So these are the findings, that two putative BAP kinases, they have been cloned, and heterologous expression it has been done, and the activity of the BAP kinase it was observed. So these are the awards received by the faculty members, and then awards received by the students, and number of publications coming out of the thesis. So total 11, and out of that one was having more than 10 NAS rating. And these are the placement of the students, and these are the trainings being organized by the department. And in the end, my sincere acknowledgements to Director IARI, Dean and Joint Director Education, Joint Director Research, Head of the divisions, faculty members, and all the students of Division of Biochemistry. Thank you very much for.
Thank you, Professor Anil Dahuja. Next presenter is Professor Debashish Patnaik, who is a professor of molecular biology and biotechnology at the National Institute of Plant Biotechnology at this Pusa campus. So, Professor Debashish, you have 18 minutes, and I will request you to kindly stick to the time, please.
Good evening, august audience, Honorable Chairman of the session, Director IARI, Dean IARI, Convener of this session, dignitaries of this dais, my esteemed colleagues and dear friends. Let me present significant educational achievements of Division of Molecular Biology and Biotechnology. A total 24 courses encompassing the whole gamut of plant molecular biology and biotechnology are offered by 26 MBB faculty members and also associate from other divisions. Total of 47 students, 17 MSc and 30 PhD students are studying in the MBB division at present. This year there are 12 degree recipients, 7 for PhD and 5 for MSc. Research work of these degree recipients could be divided into 3 broad areas: abiotic stress management, biotic stress management, and basic studies. In the abiotic stress management, research work of 1 MSc student and 2 PhD students were carried out. Mr. Jahirul, he worked to find out the role of a particular pathway, urate pathway, in salt tolerance in Oryza coarctata. This is the naturally salt tolerant species in the genus Oryza under the guidance of Tapan Mandal. He isolated total RNA from the leaves and roots of Oryza coarctata and also cultivated rice cultivar IR29 under normal and solstice conditions, converted to cDNA, and then checked amplification of partial cDNA of the gene encoding allantoids that catalyzes the penultimate step of that urate pathway. It was observed that the gene gets induced very highly in the leaves and roots of Oryza coarctata after 96 hours after salt treatment compared to that IR29, and similarly the enzyme activity, allantoid enzyme activity, and also the metabolite level allantoid, it's very high specifically in the roots of Oryza coarctata compared to that of IR29. So it was concluded that probably the metabolite allantoid is involved in conferring salt tolerance in Oryza coarctata. Mr. Rakesh, he did fine mapping of a already identified QTL involved in salt tolerance and also identified possible candidate genes present within that QTL under the chairmanship of Professor N.K. Singh. He identified 146 homozygous recombinants after screening a very large F2 population, and those homozygous recombinants were phenotyped for salt tolerance over 3 seasons, and then based on the genotyping by SNP markers, he could locate that QTL within 600 kb region on chromosome 8. And on this QTL, there are out of 48 genes, it was observed that 19 genes were expressing, and out of all these 19 genes, only 4 genes were differentially expressed under salt stress conditions. So it was thought that these 4 genes could be the probable candidate for this QTL. Ms. Priyanka she cloned and functionally validated a gene called NHX antiporter gene from local wheeled land race Khachia local, and validated function of this gene in transgenic wheat background under the guidance of Dr. Konica. The gene was found very highly induced under salt stress conditions in Khachia local, so she cloned that gene in the plant transformation vector and used that expression vector cassette in transformation of elite wheat cultivars. The putative transformants were screened for transgene integration, and then these were evaluated for under salt stress condition. It was observed that the overall physiological state of the wheat transgenic lines expressing the gene is better, as evidenced by the overall physiological growth and also higher level of membrane stability index and chlorophyll content. One MSc student and 4 PhD students' thesis work were aimed at improving biotic stress management of the crop. Mr. Norris, he studied to understand the basis of differential resistance against mustard aphids in wheat, in Brassica wild species under the guidance of Dr. R.C. Bhattacharya. He screened all those 22 wild species of Brassica for 2 years and found that the aphid population is very high in the susceptible wild species compared, it was moderately low in moderately resistant species, but very low in resistant species. And similarly, the activity of redox related enzymes were highly induced in resistant Brassica species. So it concluded that that could be reason for conferring aphid resistance in wild Brassica species. Mr. Trossand, he carried out proteome analysis in 3 genotypes upon alternaria infection, susceptible Brassica jancea, moderately resistant Synapis alpha, and also highly resistant Camelina sativa after alternaria infection. Under the guidance of Dr. Anita Grover, we extracted protein from the 3 genotypes after alternaria infection and ran on 2D gels. The differentially expressed proteins were spotted, and those were then identified based on analysis and comparison with other already identified proteins, and it was observed that these differentially expressed proteins perform these different biological functions. Out of these, the differentially expressed proteins involved in defense-related activities were highly correlated with the resistance level of all these 3 genotypes. Ms. Anupama validated 2 novel Bacillus thuringiensis genes for its efficacy against polyphagous insect pest Helicoverpa armigera under the guidance of Dr. Sarvjit Kaur. These 2 novel BT genes were codon optimized for plant expression, and these placed on before the for constitutive expression, and these vector cassettes were used for tobacco transformation. The Cry1 AC gene was introduced into the transgenic tobacco by agrobacteria mediated transformation. The putative transformants were screened by PCR and RT-PCR, and it was observed that 66 to 95% larval mortality of Helicoverpa were observed when larvae were fed on the detached leaves of selected transgenic lines overexpressing Cry1 AC34. Similarly, the mortality rate of, in case of tobacco transgenic lines expressing VA44 was found to be 45 to 53%. Dr. Vinod, Mr. Vinod, he characterized a transcription factor called WERK33 and its role in imparting a tolerance against alternaria under the guidance of Dr. Anita Grover. The gene was cloned from a resistant species called Camelina sativa and found that this gene was highly induced in Camelina sativa upon infection of alternaria. So he cloned the gene, sequenced it, and analyzed its in silico analysis. The gene was cloned in plant transformation vector and introduced into Arabidopsis, and the transgenic Arabidopsis expressing this gene showed very less susceptibility towards alternaria infection. Ms. Sikha, she studied the response of 3 different genotypes, Brassica jancea, Synapis alba, and Camelina sativa upon alternaria infection, and also exogenous application of salicylic acid, jasmonic acid, and also AVA at pathogenic, histological, biochemical, and molecular level. Based on her study of pathogenicity screening of different quaternary isolates, she could find one particular isolate ABSP2 which was more virulent on these 3 hosts. The jasmonic acid level was found very high in resistant genotype Camelina sativa, which was an indication of jasmonic acid mediated defense response which mounted upon quaternary infection. But in contrast, salicylic acid level was found very high in susceptible genotype Brassica jancea, which is an indication of a defense pathway which is very ineffective against that necrotop. Research work of 3 MSc students and 1 PhD student was of basic in nature. Mr. Mohammed, he cloned and characterized gene encoding wheat nitrate transporter under the guidance of Subodh Sinha, which could be utilized for nitrate utilization efficiency of this crop. Based on the Arabidopsis gene sequence as reference, he could clone 2 homologs from wheat NRT 1.5A and B present equally on all 3 subgenomes, and did phylogenetic analysis and found to be the grouped at 2 different groups, and genome organization of these 2 homologs are found to be very different. These 2 homologs are found to be equally expressing in D and A subgenome. Mr. Johan, he characterized promoter of a wheat gene called PM19 that encodes a plasma membrane protein. 4 different deletion fragments of the promoter were PCR amplified and placed before the gas reporter gene for its transient expression. When these vector cassettes were vacuum infiltrated into Nicotiana benthamiana leaves for transient gas expression, it was observed that the 2 promoter fragments P1 and P2 could drive gas expressions equally well or consistent level at different treatment levels. So it was thought that a particular region of the promoter could be responsible for driving expression of the PM19 gene. Mr. Krishnan, he attempted overexpression of acetylcholinesterase of Helicoverpa armigera and tried to do characterization of this recombinant protein. Although partial fragments of this cDNA could be amplified, but the full-length ORF and also the 5' UTR region of the cDNA could not be amplified despite repeated attempts. And that this bizarre result is very inexplicable, and it was thought that could be high sequence divergence and also very high GC rich region presence at the 5' end of the ORF could be the reason for this non-amplification of the ORF of the gene. So 5' race strategy was employed to solve the riddle. Ms. Antora, she studied the role of a particular class of regulating non-coding RNA called non-coding RNA in seed and pod development of pigeon pea. She carried out RNA-seq analysis from the tissues collected at different stages of seed and pod development, and based on the RNA-seq analysis, she could find out more than 3,000 LNC RNAs. And based on the differential expression of these LNC RNAs, it was observed that about 1100 LNC RNAs are showing differential expression. Then she analyzed relative expression of few selected LNC RNAs and their target mRNA. Her study revealed that this particular regulatory class of RNA could regulate the mRNA of the target mRNA involved in seed and pod development in pigeon pea by regulating as or operating as endogenous target matrix of another regulatory class RNA called miRNA. I am happy to inform you that majority of degree recipients are well placed in out of 5 MSc recipients, 4 MSc recipients are pursuing PhD degree in IARI and 1 MSc recipients are pursuing PhD study in NCBS Bangalore. 3 PhD recipients are now serving ICR as ARS scientist. 1 is working as state agriculture inspector. 2 PhD degree recipients are working as research scholars in different institutes and universities. Out of these 12 degree recipients published a total of 13 research papers of different NAS ratings journals, and MBB faculty published a total of 76 research papers in 2018 and 19 of different NAS rating journals. It's a very happy moment that Ms. Sikha, she has been selected for IARI Merit Medal and will receive this IARI Merit Medal this year. Apart from that, Mr. Mohammed has been a member of this group that received Best Oral Presentation Award in National Conference on Integrative Plant Biochemistry and Biotechnology. The honors and awards received by the faculty members of MBB is a reflection of their excellence, and I'm happy to inform that Dr. R.C. Bhattacharya, he became Fellow of the National Academy of Science, India. Dr. Subodh Sinha received fellowship to undertake research in UK. Dr. Nimi and Dr. Sondha received Best Oral Presentation Award, and also Dr. Sondha received Young Scientist Award. Apart from this education, the center is also involved in HRD, imparting HRD training to other faculty members, and the center conducted 2 training programs and also arranged special lecture programs. Out of that, 4 lectures were delivered by foreign experts. These are the glimpses of students doing different practical classes, and the happy faces are indication of their satisfaction in learning. The division also encourages extracurricular activities by the students. These students are showing their extracurricular talent and artistic talent during Freshers Welcome organized by MBB students and faculties, and also during Teachers' Day celebration. Last year, MBB students honored teachers on Teachers' Day. With humility, I acknowledge the financial support of PG School IARI and also the Institute NIPB, and also I acknowledge and convey my warm thanks and regards to Director NIPB, all the faculty members and MBB students for their support, contribution and help in compiling this information and also for presentation. Thank you all for your patient hearing.
Thank you, Debasis, for in fact making a good presentation, also saving some time for us. And we move on. We have only 3 divisions in the school, and the last but not the least is my own discipline, and that is the Division of Plant Physiology. And I welcome and invite Professor Madan Pal Singh for making his presentation, and the time is for you 13 minutes. You could take 14 if you want.
Thank you, sir. Good evening, everybody. Esteemed Professor Bansal, the chair of the session, Director IARI Dr. Ek Singh, Dean PG School Madam Rasmi Agarwal, and the Dean Convener, all dignitaries of the dais, and my dear fellow scientists and dear students. I am presenting the significant PG research achievement from the discipline of plant physiology for the year 2019. During last year, 9 students submitted their thesis, and the research work of the thesis represent the 3 important research areas of plant physiology. This is plant nutrition, 3 students, growth and photosynthesis, 1 student, and the abiotic stress tolerance, the 5 students completed the thesis. Now the first theme is plant nutrition. This is the work done by Dr. Mitra on the relationship of the iron and the phosphorus acquisition with root oxidation in wheat. His objective was to assess the variation in uptake and concentration of the phosphorus and other macronutrients and the root oxidations under individual and combined deficiency of the phosphorus and iron in wheat. He has taken 6 genotypes, the 4 durum type, 4 bread type, 2 durum type, and there were 4 treatments with different combinations of the iron and the phosphorus. And this is the data on the translocation of the phosphorus, iron, copper, and the phosphorus absorptions in wheat. They observed that under the deficiency of the phosphorus, there was a reduction in the transfer index for the P, but the phosphorus use substance increased, and this was irrespective of the presence of the iron. Then the translocation of the iron decreased, but the copper partitioning improved under both minus P and minus Fp conditions. Then this is the root oxidation and the zinc manganese uptake in the wheat genotypes, it was observed that under the deficiency of the minus P and minus iron, there was highest oxidation from the roots, but under the plus conditions of the both iron and the phosphorus, the oxidation from the roots was the lowest. The P deficiency improved the root zinc uptake under both the plus P, plus and minus Fp conditions. Manganese level significantly increased in shoot under minus P conditions, but increase in roots under both minus P and minus Fp conditions. So he concluded that the phosphorus and iron deficiency can induce release of organic acids and phytonutrients, which may facilitate the uptake of the iron and other macronutrients in wheat. The another work done by Jagdishan under the guidance of Dr. Lakshmi, the objective of his work was physiological molecular analysis of nitrogen use absence in rice genotypes and identify the genes encoding the NLP transcription factors. Then this is the work on physiological molecular analysis of the rice genotypes. They found that based on the nitrogen absence and the nitrogen responsiveness, they identified 4 genotypes, and among them the genotype IR3-11, it showed the high expression of nitrogen uptake genes, and which was followed by the Pusa Basmati 1. Also the same genotype IR311 showed the highest expression of all the LETS genes, including the RT1.1b. They also identified 6 NLPs from the rice or rhiza sativa, and the genes were named as NLP1 to 6 based on their similarity to the Arabidopsis NLPs and previous reports in the rice. Then this is the expression analysis of NLP genes under the nitrogen stress conditions in rice genotypes. They observed that NLP showed maximum expression in the roots but not in the shoot. Also the NLP 1, 2, 3 and 6, they showed the more expression in the roots. And the genotype IR311 showed the upregulation of the NLP expression, and they concluded that the positive correlation of the expression of NLP genes with high level of the nitrate uptake and acid-based genes suggests that it could be a potential target for improving the nitrogen use efficiency in rice. Another work done by Vasundhara for her PhD program under the guidance of Dr. Bhupender Singh, and the overall objective of her work was to determine the interactive effect of the iron and sulfur application on micronutrient uptake and translocation and release of the phytonutrients. She has taken the 4 genotypes, 2 bread type, the 2 durum type, and there were 6 treatments, 6 combinations of different iron sulfur application. And this is the data on the micronutrients concentration in the roots and shoot, and it was found that the sulfur application enhanced the uptake of the iron and sulfur and availability of the more iron, and also it improved the shoot zinc but decreased the plant manganese content. On the other hand, the iron application reduced the zinc and manganese content. This is the and also the application of the iron, it enhanced the concentration of the 2 important sulfur containing amino acids, that is cysteine and methionine, and it was found particularly in the bread wheat genotypes. These are data on the release of phytonutrients, and it was found that the content as well as the release of phytonutrients both markedly improved with the application of sulfur under the minus iron conditions as compared to the sufficient iron conditions in bread wheat genotypes, and similar response occurred at all 3 stages in the plants. This is the effect of sulfur application on the sulfur translocase index and the iron uptake, and it was observed that the increased supply of the sulfur showed the reduction in the sulfur translocase index but improved the iron uptake under both plus Fe and minus Fe conditions, and it was concluded from the work that sulfur application can improve iron uptake and availability, zinc content and release of phytonutrients under normal as well as iron deficient conditions in bread wheat. The next thematic area was the growth and photosynthesis. Here the Ravindran has done in his MSc program under the guidance of Dr. Rakesh Pandey, and the objective of this work was to explore the variability in growth and photosynthesis related traits in the set of 230 rice Nagina 22 mutant population. They have taken the all complete population in the field conditions, and based on their growth response, 38 contrasting type mutants were selected for the photosynthetic analysis. This is the data on the variability for the dry matter production, and there was found that there was a large variability, and the higher photosynthetic capacity in terms of the NAR in the N20 mutant population was significantly related with the leaf thickness. This is the data on the rate of photosynthesis in 38 genotype. You can see the large variability is there. The rate of photosynthesis varied from 12 to 38 micromoles per meter per second. And also, if you see the lowest value is there in the in mutant M57, while the other mutant SM67.1 was showed the 48, and in the middle, the green bar is of the normal type, the wild type, the N22 mutant. And also, the AICR curve was drawn, and it was found that the high rate of carboxylation was there in the mutant, which showed the highest rate of photosynthesis compared to the lower one. And then the in vivo Rubisco carboxylation rate was analyzed on the basis of leaf area as well as the rate of in the soluble proteins. It has found that the mutant which showed the highest rate of photosynthesis on leaf area basis, it showed the higher carboxylation rate, but the same rate of carboxylation when it was analyzed on the basis of the soluble protein, it was found lower. So it suggests this mutant SM67-1, it showed the high carboxylation. Initially, it suggested the possibility of the more Rubisco efficient enzyme. However, it was due to the more leaf protein available in the leaf rather than the true Rubisco efficiency. And it was concluded from this work that high rate of photosynthesis in these N20 mutants was due to the more Rubisco content rather than availability of the more Rubisco efficient enzyme. The another third thematic area of the research is abiotic stress tolerance. And the first work done by Mr. Pradeep for the MSc program under the guidance of Dr. Vijay Paul, and his overall objective was to assess the photosynthetic contribution of ER and ONS in the yield of heat under the heat stress condition by late planting. And they have taken the 29 contrasting weed genotypes which differ in their ON and ER size, and the heat stress treatment was given by late planting conditions. And this is the picture of the experiment. And they observed the contribution of the main shoot, ONS and the ears in the yield of main shoot in the wheat, and it was found that the ear contribution, it was positively correlated with the yield, and it ranged from 1.9 to 45%. On the other hand, ONS contribution ranged from -12 to 30%, and this negative contribution in some of the genotypes, it suggests that the ONS, in these genotypes, the ONS may act as sinks rather than the source. And then they also measured the tissue temperature of the main ear during the grain filling stage by using the IR thermal camera, and it was found that the tissue temperature ranged from 28 to 35 degrees Celsius when the air temperature was 35 degrees, and it showed the negative correlation in the main ear yield. And they concluded that both ear and ONS, they contribute significantly to the wheat grain yield, and the contrasting genotypes can be explored in future wheat breeding program for late planting conditions. Another work was done by Purushottam under the guidance of Dr. Pramod Kumar, and his objective of this work was regulation of the enzyme cycle activity for improving heat stress tolerance in wheat under late planting conditions. They have taken 2 wheat genotypes, given the main treatment of the heat stress by late planting, and the sub-treatment was the foliar application of some of the regulators of the xanthophyll cycle. This is the data on the photosynthetic pigment profile and the composition under heat stress. It was found that in both the genotypes, the level of the xanthophyll content increased with the application of these activators, and that is the ascorbic acid and also the calcium nitrate, and the level was lower when the inhibitor DTT was used. Similarly, the response of foliar application, the membrane stability index, lipid peroxidation, the membrane stability was improved and lipid production was lower, as well as the expression of the VDE gene, which is mainly involved for the biosynthesis of the xanthophyll cycle, was improved by the application of these activators. And then this is the effect of the xanthophyll cycle activator on photosynthetic and the rate of grain yield. You can see in both the genotypes, the rate of photosynthesis was improved under the heat stress condition with the application of these chemicals, and also the heat tolerance index was improved, as well as both the genotypes, they showed the improvement in their grain yield with the application under the late planting conditions. So the study concluded that xanthophyll cycle activators can reduce the heat stress tolerance in wheat genotypes through enhanced level of the xanthophylls and the changes in various physiological traits like membrane stability, rate of photosynthesis, etc. This is the work done by Wasem under the guidance of Dr. Anjali Anand, and his objective was to analyze the sugar biosynthesis and partitioning under the salinity stress in contrasting sugar beet genotypes. He started the work with screening of some of the genotypes of sugar beet, then he identified the 2 genotypes as moderately and the 2 tolerant for the salinity stress, and conducted the 2 experiments, one in the hydroponics for the short-term exposure to salinity, another was in the long-term conditions in the pot culture, and they found This is the data on the partitioning of the 14 carbon, and it was found that the translocation of 14 carbon was mostly to the newly developing leaves, and it may be used for production of the photosynthesis and the subsequent storage in the roots, particularly in the monotolerant genotypes. This is the sugar profile in the leaves and the roots, and you can see that compared to all the sugars, the raffinose content was higher in the tolerant genotype, suggesting its major contribution to the imparting cell tolerance. This is the data for the long-term experiment, and the similar response was observed in both the genotypes. And this is the sodium-potassium ratio. It was found that there were high partition of the sodium towards the leaves in both the genotype, which resulted in a high sodium-potassium ratio. It might be imparting the tolerance to the osmotic adjustment. And they concluded this is the model that the raffinose content the leaves and store in the sucrose in the roots may be the important trait for maintaining the osmotic, protecting the cell membrane in the salinity conditions. There is a work done by Duha in the PhD program under the guidance of Dr. Chinaswamy Viswanathan, and her objective was to analyze the nitrogen uptake and use efficiency of wheat genotypes under the water deficit stress, and to study the crosstalk between the nitrate and AB signaling under the water deficit and nitrogen deficiency stresses. 3 experiments were conducted. One is the hydroponics initially, then the evaluation in the field condition for 2 years, and the third one was the in 2 genotypes regulation of the gene involved in nitrate signaling, uptake, and assimilation under the dual stress. And there are the 4 treatments: the control, low nitrogen, osmotic stress, and the combined stress of nitrogen and drought. And this is the data of the first experiment, the clustering of wheat genome. What they observed, the root surface area, dry weight, and shoot were the, and seedling biomass were found the most contributing traits under the combined stress. And based on the stress susceptibility index, they identified the 4 wheat genotypes as the most tolerant one. Then this is the data on the wheat evaluation. 12 genotypes representing the all 4 clusters were selected as efficient, moderately efficient, and moderately inefficient as well as the inefficient. And the mean value of the nitrogen efficiency for biomass under the control and all the treatment, it ranged from 66 to 96 grams per gram biomass. Similarly, the nitrogen use efficiency for the grain yield also varies from the 28 to 42 grams per gram. And uptake, drought stress reduced the nitrogen use efficiency for biomass as well as for the grain yield significantly while under the combined stresses, and 3 genotypes showed the higher nitrogen use efficiency. Then these are some of the conclusions that drought stress can reduce the nitrogen use efficiency for biomass as well as grain yield significantly, but combined stress can mitigate the adverse effect of the drought stress on energy. Function analysis of the osmotic stress regulated genes in the for nitrogen response will help to enhance the tolerance to wheat to combined stresses, and the genotypes identified for the high nitrogen use efficiency under the individual and combined stress can be used as donors to develop the mapping population for QTL mapping and molecular genetics. And this is the last presentation done by the work done by Guru Murthy under the guidance of Dr. Ajay Arora to evaluate the diverse group of wheat genotype for the stem result mobilization. They have taken 43 genotypes, gone under 4 treatments. This is data for the SRM, and they found 6 genotypes with high SRM, and that was they found that a high SRM was associated with the high level of the fructose biosynthesis. This is the correlation of the stem specific weight and the SRM, and this is the correlation of the harvest index, and they found that the SRM showed good correlation with these traits. And the conclusion was the SRM can be considered an important trait for screening the wheat under the different stresses, and wheat genotypes which are identified with high SRM can be used under the combined heat stresses in the future climate change program. These are the student placement, done PhD, and 2 are in ARS, 2 are the foreign student already in service one. 2 training programs are organized. The one of the student got the Khurana program scholar for 2019, and he has also been selected as the ASBP scholar for 2020. And then the 4 students were awarded the CAST fellowship. 2 students got the DBT Newton Bhabha fellowship this year. And this is the best poster award given to 2 students at the National Conference of Plant Physiology. And the 2 of our students get the 2nd prize in the science quiz organized during the National Conference in Thrissur. Thank you very much.
Okay, thank you. Thank you, Dr. Madan Pal, for a good presentation and keeping up the time almost. You were there. And now I believe I have this opportunity to thank all the professors of 3 different disciplines of this School of Basic Sciences. And I'm sure students have got benefited, including the faculty members from within and outside the division of School of Basic Sciences. Hand over to the organizers for the further proceedings, please.
I request you, sir, for your valuable remarks. Just a few minutes.
Okay. Well, I will not be able to go discipline-wise if I want to make a summary, or even student-wise from the 3 disciplines or divisions. However, what I have observed in the work which was presented by the 3 professors, that there is a great deal of commonality of the kind of research which is being carried out by this School of Basic Sciences. I'm first of all happy that these different schools have been organized and presentations are being made school-wise, while we have several disciplines. If I remember, there were 23 or even more now across the PG school disciplines if we talk about it, but it's a good system and pattern to have these presentations school-wise. So when I see what the things were common, you know, and I come from basically myself a plant physiology division, then my minor was biochemistry, and as Professor Ashok Singh mentioned, I learned genetics from him, and then I ventured into the area of plant biotechnology. So I cover all the 3 disciplines, actually speaking myself, while being a practicing scientist, and that too right here at IARI. So what I find, therefore, kind of common, you know, common areas which I have, for example, identified, or you all know, is this, that one is that all the 3 disciplines are working on abiotic stress tolerance, whether you talk of biochemistry or physiology or, you know, plant physiology or biochemistry or biotechnology. Second, that of course after abiotic stress there's always a lot of emphasis on biotic stress tolerance, and I see that this is also being covered by all the 3 disciplines. And third is nutrition enhancement is given a focus, rightly so, by division of biochemistry, but I see there's a lot of potential, at least when we talk of a discipline of molecular biology and biotechnology, to also, you know, give their own emphasis on nutrition enhancement when we talk about it. And fourth is developmental studies, which were of course restricted to few theses, you know, particularly in the area of molecular biology and biotechnology. But most important area which I have pleasantly seen in these presentations was, actually speaking, while we have been working, I know all these areas are important, but with time where there's a possibility of seeing some good results in the form of a product, is actually speaking nutrient use utilization efficiency. I'm very happy to see again across disciplines that there is a very good emphasis given here by division of plant physiology, also biochemistry, and I could see even in biotechnology as well, that there is a lot of, you know, emphasis given on these subjects. And another common factor which I found was the kind of methodology or tools we are utilizing. What I talked to you was these science areas we're talking about, of disciplines. Within a discipline, what are the focus areas? But then all these 3 disciplines, I find that there is a common use of various tools or even material. When we talk of material, what is being utilized, I have seen, is use of these increasing use of wild species. When you talk of brassica, or you talk of wheat, you talk of, or land races, you know, for that matter, not only wild species, or land races, also very important for understanding the basic physiological mechanisms or biochemical means, or utilizing the genes and alleles from some of these land races for improving our cultivated crop plants, which are otherwise very high yielding, but they do have, you know, possibility of adding into them the resistance to biotic abiotic stresses. Particularly increased use efficiency of nutrients and natural resources is very, very important, and I'm happy there's some emphasis given on that as well. And when we talk of tools which are commonly utilized, I found that, of course, again, across the 3 disciplines, because I see here, you know, we're talking of genes, understanding the expression level of those genes under various conditions or stages. We're talking of gene cloning, we're talking of developing gene constructs. Now also we're talking of constructs with guide RNA sequences, etc., using CRISPR tool as a common tool again. I find that's also being done almost in all the 3 divisions. Dr. Viswanathan started this with divisional plant physiology. I'm happy in soybeans being worked out in biochemistry division, and of course again in rice and many other crops. You know, we can see there's a potential, you know, there in case of division of, you know, molecular biology and biotechnology is also being taken up in a big way. Though there are very many efforts not there in the country at the moment, but I think this is important that we are moving towards that goal. But if I come to my general observations and points, what I find as I have been watching, that there are students here, faculty members here present from various other disciplines, and I was told that Professor A.K. Singh, our Honorable Director, made a very important strong point yesterday that we must have the presence here in this hall for either the students' presentation yesterday which were held, or even today, for professors' presentation, they should all be there. So accordingly, our professors from different disciplines, I am addressing at the moment to the professors of these 3 disciplines, they must try to make it more easy to understand kind of a presentation. Not everybody will understand gene cloning, not everybody will understand the kind of nuances of these basic sciences, but I think while concluding, or while even giving some kind of objective and goals of various experiments, concluding all together that this particular discipline added some kind of value or knowledge generation in this area for enhancement of what next. I think in a very general, common, popular understanding, it's very, very important. And then while I congratulate all the students, they're doing very well, and I of course know through the hard work of the faculty members, they're doing very well. But again, in an agricultural setup like this is the only institution we have in the country on agriculture. We have such a large national agricultural research system, you all know, with 100 and more of ICR institutes and 75 state agriculture universities. No other institution will have that kind of faculty member and strength in basic sciences as much as we should project and have more in an institution like IARI. So what I believe and look forward to is that we should have a discussion on what is that kind of a new approach or innovation we are able to add, and really speaking, give more and more emphasis on basic sciences. Whether that's going to be translated tomorrow into a product or not, irrespective of that, I believe it's important when we call it as a school of basic sciences, there should be a more ingredient of basic sciences and innovations into it. I think that kind of a discussion is very well required, that what is that new knowledge can we generate and add to the international literature. And then I think this is possible again, that while there are common areas of interest, there are common tools being utilized, can we think of an innovative interdivisional or a school-wise or a basic sciences school project, wherein the faculty members from all the 3 disciplines will contribute effectively, but that has to be not just kind of run-of-the-mill experiment or a project, it has to be something again innovative. And you have to have an external funding for it, not that it should be coming from within IRI. I know funds are always a constraint. So that should not be that, okay, because we don't have funds from IRI, we are not able to do this project. I'm sure there are very many funding agencies across the world that we can attract some of these funds. And that should be there. And then again, what I have in my store, only a few more points, that another general message I would like to give, while these disciplines of life sciences, if I call it in general, when we're talking of microbiology, you're talking of genetics or plant breeding, and also biotechnology and basic sciences, what we should remember, and that the world productivity, when we're talking about the global level, if we want to increase 100% productivity, 77% of it, as experiments have shown, will come from genetic gains, from the seed which you will give to the farmer. 23 will come from the efficient utilization of natural resources, or whatever management of biopesticides or anything you want to do, the contribution of is going to be, as has been projected globally, 23%. So therefore, more and more emphasis in an institution like this to be given either through basic sciences or through other disciplines of life sciences and agricultural sciences on increasing the genetic gain, including the involvement of plant genetic resources, because that's also one of the divisions of the PG school here, utilization of in what way or the other, as you've been already doing it, the kind of land races. It's very important that you develop a new seed. As you would have seen, the kind of, kind of varieties which have been developed in rice is an example to talk about it in the world. But still, I think a lot need to be done when people taking away genes from our own material and utilizing, for identification of genes and alleles, and then we, try to introgress into our own, cultivated types. So I think that that's very, very important, this kind of a genetic gain to be having it more. And this is my final remark, and that is, while you are not working directly with the students anymore, and when you have left the institution like ICAR and you're retired, you always try to think what more could have been done. What I believe, while I've had about 20 PhD students with me, But then, and we worked on different projects, and so you all working even more, projects through your students. But I believe it's our responsibility again, in an institution like this, with PG school having such a long legacy of at least 62 years now, an institution having more than 100 years, it's important that we produce our PhD students as thinkers, not only as degree holders or subject specialists, as thinkers, critical thinkers. Really speaking, that they can stand on their own and they can develop a new project altogether without your hand holding further. I think that is something very important. What parameters can we have it in this direction? One has to think about it and accordingly devise your course curricula. Across disciplines, we have subject minor and major disciplines, but I think this is something that we need to give more emphasis on, to produce thinkers, critical thinkers. Particularly, I'm talking PhD students, not as much emphasis on master's thesis. So with this, friends, I express my gratitude. I'm extremely happy to be here to give you this kind of, give me an opportunity for interacting with you all. And finally, I express my gratitude again that I'm sitting next to your current beloved director, Professor Ashok Singh. And thanks to all the audience, to the students, to the faculty members, and to the professors who made this presentation on basic sciences. I wish you all the best. I congratulate all the students who got awards, who are going to get degree also on 14th of February. Thank you again. Thank you all of you.
Thank you very much, sir, for bringing key insights from this session. We are charmed and charged by your visionary remarks. May I now request to the Director IARI, Dr. A.K. Singh, and Dean and Joint Director Education to kindly do the honor of presenting a shawl and memento to the chair of this session. And I request the convener of this session for vote of thanks.
It is my privilege to thank Dr. K.C. Bansal for nicely summarizing the work of disciplines of basic sciences. And this, I'm sure that his suggestion will be well taken by the faculty and the professor of the basic sciences discipline. And not only this discipline, some of his suggestions are valid for all of us, and probably we will take care of this in our next presentation while preparing the presentation and looking after all his suggestions very well. And now I think, no sir, it's nice, thank you very much. And now we will, I think, start with the last school, the School of Horticultural Sciences, and we will be having, and I will request everyone to be seated here, don't move please, so that you will also be benefited by the four professors' presentations. We request next chairperson, Dr. D.P. Ray, to be on the dais, please.